Okay. Thank you so much, everyone, for joining us today. We have the pleasure of hosting Dr. Rosengarten, if I pronounce your name correctly. I let him introduce himself, but briefly highlights of his background, if I want to mention. He's a Yale graduate, and he has done his second postdoc at Baylor, so he's considered as a local. He'll be talking about like his experience, his career path, and his current company, and the openings they currently have, and also the opportunities they are planning to provide in the future for the scientists in the area. So please welcome him to GSBS by just giving him a hand. Okay. And 2 points, please write your name if you may, and your institute's name as well. Also, for your information, we are recording this event, and we will be putting it on YouTube. So if like anyone has problem, you won't be in the recording. Of course, it will be only him and like in that monitor. So please stay like in this area.
That's actually super distracting up there.
Don't look at yourself too much. But thank you so much, everyone.
Thanks, Sina. Guys, thanks for coming out today. I wandered over here a couple weeks ago because we were recruiting, and I wanted to find talent, and this seemed like a good place. And Sina invited me to give a talk, and I'm excited to do that because not only is Genialis, my company, looking to hire one person now and many people in the future, and we'd like to build locally, but we're also really interested in giving back to the scientific community in Houston. Not so much giving back, but working with, becoming a full-fledged part of the community in Houston. Already we work with Baylor in a number of ways. They're a commercial partner, but also a development partner. And so we'd like to kind of expand our reach around the medical center. Today I'm going to give a presentation sort of in 3 parts. The first 2 parts I'll throw some slides up. I'm going to just give a bio. I have a very windy career path thus far. It's definitely not an A to B sort of thing, and so I'll talk about it a little bit, just to illustrate how, in my opinion, that's how life is led. You kind of follow your passions and jump at opportunities. With that said, some of the most successful people I know, knew from the very beginning exactly what they wanted to do, and they set out and achieved it. But I think most of us are a little bit more, I don't know, experimental or unsure or widely interested than that. The second part will be to talk about my company. It's not meant to be a sales pitch, but if you guys want to buy some software, my business cards are right up here. But it's describing our mission, and to see if it's something that excites you guys, or maybe you have some friends who would be excited to participate, either as users or as employees. And then the third part, we can leave the slides alone, and I want it to just be a really interactive sort of Q&A. I'm going to ask you some questions, you can ask me some questions, and that is meant to focus on general strategies for building a career and getting a job. You can ask me about anything I've shown in the slides, but we can also talk about how to use LinkedIn and what's the difference between a CV and a resume, which should be taken for granted, but I can tell you after last night's resume builder event at Baylor, 5 out of 6 people don't know. So we can talk about that. How many people here remember the moment you knew you wanted to become a scientist? Like the very moment, yeah? Or rough. What was the moment?
I mean, it was just when I was here.
Yeah. So I probably need to have a picture up here of the pine woods and salt marshes of South Carolina, where I grew up. Because I grew up as a kid in the mud. That was my playground. There was none of this, parents walking you to the park stuff that I have to do with my kid here in Houston. It was go out and run around, try not to get eaten by an alligator, and come back for dinner, sort of thing. And so I've always been in love with the natural world. I've always been inquisitive about it. But the moment that I knew I wanted to be a scientist, I was 7 years old. This is the Bay of Villefranche-sur-Mer in the south of France on the Riviera, and my uncle was director of a marine biology lab right there. And we had gone over on my first family trip to Europe, and he taught me how to snorkel. This bay, besides being kind of beautiful from an aerial view, is one of the world's most studied but also richest plankton reservoirs. It's an incredibly deep bay with a huge current that comes up from the cold and oxygen-rich depths, and so the plankton washes up, and it's just a magnificent place to snorkel. Of course, the plankton comes, and so the bigger things come to eat it. It was cold. I grew up again in the bathwater temperature ocean of South Carolina. But after my first day of shivering, they couldn't drag me out of the water, and I knew I wanted to be a scientist. My uncle comes from the old guard where you could be a marine biologist, and that was like actually a thing you got paid to do. It's much harder to do today. There's not as much funding for it comparatively. But I was hooked. So the first time I got paid to do science, I was in high school, and I had to do a summer internship as part of my graduation requirements. So I got a job at the Medical University of South Carolina in Charleston working on embryonic chick hearts. I didn't have a particular interest or affinity for developmental biology per se. I still thought I liked marine critters better. But it was a neat chance to get in a lab. They had an early confocal microscope, so just to date myself, this was 1995, 96. They had one of the first silicon graphics desktop units to analyze that. And I showed up at the lab, and the PI was out, and he left a post-it, "Go play on the silicon graphics computer." I guess he assumed I was young, I knew what computers were about and how to use it. So that was a neat, interesting summer, just kind of getting my hands dirty. Incidentally, the guy I was working for in Charleston, he actually grew up in Texas and still has a beach house in Galveston. I went to Dartmouth for undergrad, and when I was at Dartmouth, I thought I was going to be a chemist. At that point, I had really fallen in love with like hard science, where you needed to know math and think about things on subatomic scales. I liked physics a lot. Either I got lazy in school, or I just kind of got re-inspired by nature, being up in the great North Woods. But I fell in love with ecology and evolutionary biology. I had a great physiological ecology professor where we would think about physiology and think about chemistry, but we'd walk around the woods all day. And that was cool. That reminded me of my childhood. I did actually a winter program because we were in New Hampshire. It was a better time to get out of Dodge in the winter. In Costa Rica and Jamaica, studying tropical ecology. And when I was in Jamaica, on the coral reefs, I came up with a question that was sort of the first real scientific question that I felt like I needed to go out and answer myself, besides just being curious. And I was interested in how two organisms on a crowded reef know who the next door neighbor is. How related is a reef to itself? And it turns out there was a guy at Yale who was doing just that, and so I was going to go to work for him. I actually did right out of college. I got a job at Yale in his lab. But it was around this time that I also was thinking, if I'm going to go to grad school, if I'm going to be a scientist, and this is what I'm going to do, that's a big commitment, and I'm kind of scared of commitment. So maybe I should, go play with some of my other hobbies and interests first. Just give them a try. In college, I had started cooking a lot. I thought maybe I'd want to be a chef instead. So I did that. I dropped out of science for 2 years, year and a half. I went into New York City. I started in Tribeca, and walked 65 blocks north to about the bottom edge, a little into Central Park, and I dropped off 20 resumes at the top 20 restaurants in Zagat in New York City. 3 of them called me back. The first one I went to for a tryout offered me a job, and I spent the next year or so before grad school working at Michelin. It's closed now, but not before it earned 2 Michelin stars. I mean, this was like, this wasn't Thursday night in Hoboken, as we used to say. This was like serious food. So I did this for a while, and I loved it. It was actually the funnest job I ever had. If I didn't care about having girlfriends and real friends and exercise and seeing daylight, I'd still be a chef. It was the best thing I had ever done. I regret every day not continuing with it in some small part of the back of my mind. It's also the hardest work you'll ever do. The service industry stinks. It's miserable. It literally will ruin every relationship in your life except with the bartender and the barback. I mean, but I loved it. Anyway, I did go back to grad school. I went back to Yale to work in the lab that I had teched in for a while, still interested in this question of how do you know how related a coral reef is to itself. But life throws curveballs. The PI whose lab I had been running as a technician had gotten really sick, so he wasn't taking grad students because he told me he had between 6 and 9 months to live. He is still alive today. He's actually doing quite well, but he wasn't taking grad students. He's actually retired. So I went to work for his collaborator who was in the cellular and developmental genetics department or something like this, which was really good for me actually, because I had, in college taken this turn towards what I consider a slightly softer science, at least a more anecdotal science in ecology. And so going into the molecular biology program, the genetics program, forced me to actually learn molecular biology and genetics. Here's a embarrassing admission. I've never studied organic chemistry. I have never taken an orgo class. I've only taken one physical chemistry class. I am grossly underqualified to be like a proper cell biologist or geneticist, in my opinion, because I don't understand things at a smaller scale, or at least I haven't studied them. But I went back to Yale, and in the collaborators lab in molecular genetics, I had studied these two organisms, which are highly related to reef-dwelling organisms, and I figured out how they recognize how closely related they are, how they mediate the decision between whether two distinct tissues, either allogenic or exogenic tissues, are going to fuse or reject. And this probably resonates with anyone here who's studied immunology and transplant biology. So we use this as an interesting sort of bizarre evolutionary model for how multicellular how animals can do self-non-self recognition. It was a really, really cool system, a really hard system to study. We didn't have a genome at the time. So I learned lots of techniques like building back libraries, Sanger sequencing. I spent 9 miserable months teaching myself Perl so I could analyze those Sanger sequences. I actually studied at Cold Spring Harbor for a couple courses to learn how to do that. But it was neat. So I was simultaneously learning how to do what I consider really interesting and hard and relevant, sort of potentially biomedical science, but on a cool, funny organism. I actually got to go scuba diving a good bit. We would go to the Caribbean, not to collect these organisms, but another organism I worked on. These guys just lived in the Long Island Sound, so that wasn't so much fun. Until I learned how to culture the organisms in the lab, and then our Caribbean trip stopped and everyone hated me. By the time I graduated, so my last year of grad school took 2 years, and by the time I was done with it, I had all of a sudden gotten interested in something else. At this point, I had met and dated and broken up with and started dating again the woman who's now my wife, and she had landed a job with Chevron, which is why I live in Houston. And so I was thinking about fuel and where people do energy work and where my life, she was always going to make more money than me because she worked for a real company and I was an academic. So I was like, all right, well, I'm going to be following her, so I better do something where I can get a job too. I didn't actually realize that Houston had this whole research complex. I just figured I'd be stuck in energy. But my brother at the same time, he was getting into environmental policy stuff. And so I was reading a lot more about sustainability, just kind of tagging along with his interests. And these things, there was a confluence at biofuels. Congress had just funded the Joint Bioenergy Institute as a division of the Lawrence Berkeley Lab in the East Bay of Bay Area, California. It's actually in Emeryville. And I went out there. Actually, I had a phone interview. So there's going to be a lesson in this. I had a phone interview with a woman who was running part of the fuels division, developing, it was microbiology basically, developing yeast strains that could make gasoline. And the interview didn't go very well on the phone. She passed, gave the job to someone else. But I was heading out to California anyway, so I figured I'd just stop by and thank her in person for the interview. So I did, and after about 20 minutes, she said, I'm not hiring anyone else right now, but another one of the directors down the hall, he is. They're starting up a synthetic biology group, and they need a postdoc. 2 hours later, I walked out the door with my postdoc. Yeah. So I don't know, the lesson is, just shove your foot in the door, I guess. Human connections matter is another part, especially in the world of LinkedIn and teleconferencing and cell phones. Human connections I actually really think matter, and I'll talk about that more about software sales. Anyway, I got involved with this synthetic biology group at the Berkeley Lab, and oddly, I started working sort of on a software project. I was actually in the lab, but I was validating the results and the workflow of the software we were building to help biologists do cloning better. So we wanted to build plasmid libraries to overexpress proteins in E. coli that would make gasoline or diesel fuel or whatever. But we had no idea of knowing the best combination of these enzymes, and so we were building plasmid libraries with like 25,000 plasmids in a tube. And we had to do this 6-part combinatorial cloning, and it's hard to design, it's hard to pull off in vitro too. But we were working on software, and this software still exists. It's free to use. If you do a lot of cloning, you can check it out. It works. It's definitely academic software, but it works. But I kind of got this bug while I was at JBay. Because Congress wrote the sort of charter for it in a certain way, we were at JBay, the institute was graded on 3 things. First was, how many companies can you spin out? Second was, how many patents? Third was publications. So sort of the opposite of a typical research lab. So lots of people were interested in starting companies, and I kind of got this commercialization bug. First time in my life that I ever actually cared to do something that impacted people that weren't coral. So this was weird for me, but it stuck. Anyway, my wife's job moved from Chevron headquarters in San Ramon, California to Houston, so I followed and I got a job at Baylor. I had a really good time in my 4 years of postdoc here. I really liked my advisors. I liked my lab mates. But in some ways, I think I picked my lab the wrong way. I was already, 30-something years old at this point. I didn't want to have to learn the cancer literature. I didn't want to have to learn human biology at this point, which I just didn't know. I felt like I was going to be at a huge disadvantage if I had to spend my first year of a postdoc just reading the literature instead of hitting the ground running. So I joined a lab that studied Dictyostelium, which are these cool cellular slime molds, which was like a throwback to grad school, because Dictyostelium is another model organism, a weird model organism for this self-non-self recognition thing that I already knew all about. So, I did. I hit the ground running. I think I wasn't super productive, but I ended up with like 4 papers. I made a couple actually fairly seminal technology improvements in the field, coming in as now a technologist. And about halfway through my time there, I got involved with this new company called Genialis. So Genialis grew out of a data science lab in Slovenia. Bonus points if anyone can point it out on a map, but I'll actually show you a map in a second, so guess now. Anyway, so this data science lab in Slovenia is really, really, really high-level data science, not specifically biological data, any kind of data. But they did do bioinformatics through this collaboration with my lab at Baylor, and they had built this kind of academic hackware that helped us look at transcriptomic data for this weird organism. Nothing off the shelf, really. First of all, there's very little off-the-shelf software that's good for transcriptomics, especially time courses, and there's very little that's tuned to the oddities of Dictyostelium with its 80% AT-rich genome, which causes a lot of problems both in vivo, in vitro, and in silico. So Genialis, the small company, grew out of that data science lab in Slovenia, and the first thing they did is they rewrote DictyExpress from the ground up, this software that helped us with our transcriptomics analysis. They built it in a modern tech stack, no longer Perl scripts and chewing gum, which was sort of the old one, and recruited me halfway into their project after 1 year of working shoulder to shoulder with these guys to join the company. This was about when I realized that I wasn't a good enough bench scientist to be a PI, so I thought this was a great opportunity. So again, a bit of history. That's where Slovenia is. It's in this sort of armpit between Italy and Croatia and Austria. It's actually a fairly new country. It used to be part of Yugoslavia. On the Western Front, Austria and Italy have been fighting for that land for centuries probably. On the other side, the Croatians and Hungarians have been fighting for it. And at the end of every major world conflict, one of the neighbors or more than has taken away the best parts. So Trieste used to be Slovenia, it's now Italy. The seacoast used to be Slovenia, it's now Croatia. The mountains used to be Slovenia, it's now Austria. It's still a beautiful country, but it's been whittled down to a postage stamp of really smart computer scientists. So that's where the company started. I mentioned there was this collaboration, and in 2014 they started working in a formal capacity with Baylor. In 2015 they recruited me. So my first order of business was actually to start a company. We're incorporated in Delaware. We can talk about why later, but we're based in Houston. And so the Houston office is me. It's just me. And any technical staff from Slovenia we rotate over to help service projects here. In 2016, a year later, the Houston-based company actually formally acquired the Slovenian company in a share exchange. And we did this, again, we can talk about reasons why, but it makes us an investable entity. No investor in their right mind is going to pump money into a Slovenian company because there's no case law there. And this way, we have the shares and the IP in the right place. This is our website. Maybe you've been there already, maybe not. Check it out. And this banner changes, so I actually think I did the screen grab on a message that may or may not have been the one I wanted to put up. But the point is this. Genialis has a fairly unique approach, I think, to building bioinformatics software. We're not so concerned about solving problems for the bioinformatician, although we do that. Our main selling point is we try to help the biologist, not to become a bioinformatician, but just to do more with their data. So almost all of our competitors either build software to help bioinformaticians compute large-scale data sets in the cloud or on their desktop, I guess. That's important. That's a tough problem. We've got hundreds of thousands of genomes now. How do we actually compute all that at once? We're not solving that problem. We're piggybacking on people who do. Another problem that some of our competitors solve is how do you give sort of training bioinformaticians or even serious bioinformaticians better tools for sort of stitching together pipelines, or how can you teach a biologist to stitch together their own pipelines and stuff like that. That can be done on our software, but again, it's not our focus. Our focus is to create a really rich visual interface for biologists who again don't want to or can't learn or shouldn't learn just the sort of ephemeral bioinformatics enough to screw up their own data analysis. These are people who should be engaging with data at the level of secondary analysis to try to make sense of patterns, to try to think about, using their decades of domain knowledge, what's actually going on in the data. But we do have 4 major value propositions because we have to sell this stuff. So the first thing is we help store data. In case you haven't noticed, I'm entering phase 2 of the talk. We help store data. So some labs have either really good collaborators or a really smart sort of systems person in the lab, and they store their data in the right way. Most labs have thumb drives, hard drives stacked up, CDs burned in people's garages, maybe a Dropbox sort of thing going on, and no one knows where the data is, especially for multiple people in the lab. No one can reproduce experiments or analyses. This is actually a serious problem. 70% of papers can't be reproduced or something like that. I don't know how much of it's the fault of the bench scientist or the data scientist, but regardless, reproducibility is an issue. So we help store data so that you can actually find not only your raw data, your FASTQs and your BAM files, but you can also find every derivative analysis file. They're all linked to a parent. And so at any given point on our software, and I'll show you the interface in a moment, a biologist or a bioinformatician or your PI or an auditor could click on a sample and see exactly the analysis history from start to finish, all the inputs, all the outputs, standard error, standard out. And so that's tracked. We do have a nice work environment, both a Python software developer kit, but also a GUI for bioinformaticians to both develop pipelines and to automate them. And so the nice thing here, in principle, is with a bit of investment up front, a bioinformatician can go from being a service provider to their lab back to being the scientist that they are trained to be. So instead of answering emails all day of, why the hell haven't you mapped my data yet? Or why did it take you 3 months to tell me that the QA/QC is bad and I need to redo the experiment? You can set up the pipeline, automate that, and go back to actually working on novel algorithms or complex analyses, statistical modeling, things that need to happen but often get put off because of the sort of service nature of the just being outnumbered so badly in most research settings. Point 3 is critical. This is what I was talking before. We want non-computationalists to engage more, right? So it shouldn't be emailing back and forth Excel files to sort of collaborate between a biologist and a data scientist. We want biologists to actually be able to explore their data. If they have a question, they should be able to go answer it without necessarily having to nag someone to rerun that one analysis but with 2 new samples, or to reformat a graph axis or something like this. So we have an environment which we think, and I can tell you, having been a customer of the company and our older software, let us, idiot biologists, actually ask questions and answer them on our own. And lastly is sharing data and results. So this is key both in dissemination. Once you've published your paper or whatever, you can just change the settings on your data to public. We very often build outward-facing URLs for labs or institutes so that, let's say you're creating a new database around a series of experiments, you can now share that published data with the rest of the world. They can not only see your results, but they can even investigate them a bit. And it's also useful just for you can copy and paste the URL from within our software, email it to somebody in your lab or across the country or your PI, and as long as they have permission on the data, they can actually see exactly the state of your analysis. So this is what one of our dashboards looks like. The way we conceive of the graphic user interface. First, we have a bunch of different pages. So this is upload your data, search for your data, visualize your data, and this little flux capacitor thing here is actually the bioinformatics dashboard, the GUI for power users. And then across the top, we have, on the visualizations anyway, these tabs. And more than 5 exist, but I'm just showing 5 here by way of example. These tabs are each a dashboard, and each one consists of somewhere between 3 and 6 of these squares, of these widgets or modules. And the idea is that we really try to understand it for every customer through just, right now it's brute force interviewing, but eventually it'll hopefully become more machine learning-y. But for every customer, what are the kinds of questions you ask? And in what order when you get new data in? So very often someone will start with like a sample comparison. So, do I have batch effects or do my controls and my treatments at least align? So we offer things like principal component analysis and hierarchical clustering. But we do it in a really easy way. You'd have to run those. You'd have to at least know R or maybe some NumPy or I don't know what other command line tools there might be to do those kinds of analyses, but now a biologist can just populate their samples from the search window and they can see how those fall out. And if they see outliers, they can just strip them from the analysis or dig deeper. For example, this is a gene sets builder widget. So maybe you upload some gene sets. Maybe you've discovered some gene sets elsewhere in the app. For example, looking at a volcano plot, you highlight everything up and to the right because these are your highly upregulated, highly significant genes, and you grab those and you create a gene set. Here you can look at the overlap. So you can look at the overlap between something from, I don't know, a gene set you found from a gene ontology analysis, something from the literature, maybe something from a mouse model in your lab, and you can see the overlap. We've actually, this is a deprecated slide, we've now decoupled the heat map and created a whole new visualization around heat maps. So now the gene set builder is connected to gene ontology enrichment. We're going to build a GSEA module as well. But to give the biologist a way to say, okay, well, here are the genes that are common. And what's cool about this software is every module, every visualization module, is completely interactive. So it's all written in Angular JavaScript, which is a tough tech stack, but it makes it so you have a ton of control over their interactivity. So every mouse hover gives a pop-up or a tooltip that gives you more information. So here, the user has clicked and selected this almond-shaped intersection of these gene sets, and you get a tooltip that tells you there are 50 genes there. This click will then propagate. So this we grabbed before the propagation happened. So this heat map will now go from being these 9 genes to the next 50. And so every click here transmits here, and selections here will transmit over here, and so forth. So all of the modules are interactive and interconnected. Every click does something. It either reveals new information or triggers a new analysis. And then once you've discovered something here, so you have your 50 genes or your 9 genes or whatever, they go here, and you can take these 9 genes and you can then go look at them in a time course or in box plots or bar graphs or whatever. And so again, the idea is to let a biologist kind of follow their intuition from one question to the next until they come up with a novel hypothesis, at which point they should and must go back to their bioinformatician and say, here's a hypothesis, can we model it? We're actually intentionally very light on statistical values, so we don't give people a lot of p-values because we don't want biologists chasing them. We don't want them turning the knobs until they think they see something significant. We want them to come up with hypotheses and then ask it in a proper way. Like I said, we do have some tools for bioinformaticians, command line tools, dashboard GUI tools. In order to achieve our goal of making things easy for the non-computationalist, it takes a ton of hard work by computer people. So that's our staff, our employees, but also our partners. So for example, at Baylor, we're working really closely with a young professor there named Charles Lin. He's a computational biologist by way of Dana-Farber, MIT, and Duke. Really, really brilliant guy. He's way younger than me. He's got way more papers than I could ever dream of having on important stuff. And so we work really closely with him and his personnel, with other people at Baylor, other people at a University of Rochester and Copenhagen. It's an institute there we work with, various pharma companies, to crowdsource as much of the sort of bioinformatics backend as we can. All of the data flow engine that we've built, the bioinformatics tools that we've put on here are open source. You could actually go find our documentation for all of it. You could, if you're really ambitious, try to download it and make it work. They're just Python libraries, so you actually need to have a framework to use them. These aren't binaries. But we try really hard to engage the broader community to build out our informatics tools. This is our team. One thing is it looks very Central European. We're very cognizant of sort of the tech world in general. It doesn't have to be Central Europe. It could be Silicon Valley. It tends to be very white and very male. These are things we're looking to change, especially here in Houston, which is such an incredibly diverse city with talent from all over the world. But this is our team, and actually a couple people aren't shown here. We do have another female employee. Oh, there she is. She's on maternity leave. So no, that's all of them, 3 of them. And that's pretty good by Slovenian or, frankly, Silicon Valley standards. So we're working hard to get better at that. I actually think we've identified the rarest of rare beasts in the tech world, which is a female front-end developer. All that cool JavaScript, like Facebook and Google, man, that's a boys' club, and that's something else we're trying to shatter, something else we're trying to disrupt. So our new hires will hopefully, especially in Houston, make us improve our culture and our workplace. But like I said, I'm actually the only one in Houston. All these other nice folks are in Slovenia. Actually, this guy's here right now on secondment. He's going to rotate back, and I think our CTO will come join us again. He was here for 2 years. He's actually the one who recruited me. But we are hiring in Houston. Right now, the position we're looking for is what I call a bioinformatics software developer. So you'll be doing bioinformatics, but you're going to be doing it in a way that you build real software. Just by way of illustration, our super talented partner at Baylor, he gave us some of his bioinformatics tools, 1,000 lines of code for doing chip-seq analysis. When we handed it back to him and put it on our platform, we had whittled it down to 40 lines of code. 50-fold improvement in the code structure. That's the difference between doing bioinformatics and doing bioinformatics software. So we're recruiting for someone who's either a killer bioinformatician who wants to learn how to be a software developer, or a software developer, Python preferably, who knows enough biology or has enough interest to learn some biology to apply their software development to bioinformatics. So I mentioned we've got all the clients above the fold here are on our new software, our newest platform, which has been built since I joined the team as the product manager. Everything below the fold were clients, some of which we still engage with, others we're sort of one and done, that we engage with either as a service company or as a kind of ad hoc, we'll build you some custom software kind of stuff in the early days, before my time. But I'm excited and proud to say, and when I show this to investors, we really highlight this. There's some blue chip names on this slide, and more importantly, we've got penetration in 4 continents already. For a 15-person software startup with one full-time sales guy who is not a salesman by training, that's actually pretty good. So I wear a lot of hats, so this is going to start transitioning in the next, last 20 minutes into more of the career development stuff. In my company, I wear a lot of hats. My chief role is product manager. That means I am in charge of dreaming up what the software should do, what it should be, but also validating that that's what it should do and should be, right? Like, I can think of a million cool things to do in the realm of bioinformatics or to automate, but can we sell it? Is there a market hunger for it? Will the market support it? So it's really cross-functional. I oversee a bunch of the marketing, a bunch of the sales strategy. I do a huge amount of customer interviews. I also happen to be a project manager. So on our US projects, I'm the guy running interference with the client, but that also means I'm the one bringing back feedback on what the software needs to do. And then I manage our product roadmap. So I haggle with our project managers and with our technologists to figure out what do we build next, what's the highest priority, what technologies do we need to support the features we want, and so forth. I do a lot of corporate governance stuff. This was definitely new to me, so I spend a lot of time on the phone with lawyers, some time with accountants. I try to avoid those calls. That's interesting. What we learned early, so in the early days, the founders try to do as much of that as possible because lawyers are bloody expensive, so are accountants. Very quickly you realize that it's money well spent. It's actually the most important thing if any of you plan to start a company. The first thing to figure out, besides there's a market for what you want to sell, get a good lawyer. Someone who knows startups, someone who lives and breathes startups, someone who will do all the things correct and plain vanilla the first time. We screwed up a lot, over and over again. I try to raise money. I also do just generic business development stuff, so finding partners, strategic stuff. And we are attempting to raise outside investment, private investment. So I've had the great joy and frustration of getting to learn what pitching VCs is all about, and that's kind of interesting. I'm happy to talk about any and all of this. This is probably the coolest thing because it's on TV, but it actually is just brutal. This sounds really boring, but it's probably more important. And if any of you are thinking of starting a startup, I asked my CTO for some advice. He was the original founder. He went through the salad days. He put the first 10,000 euros came out of his bank account. That's his advice. I said, hey, Mika, what would you tell someone who wants to start a startup? Anyway, don't listen to him. It's actually a lot of fun as long as you don't care if you ever make any money or sleep. But the same is true if you want to be a cook, or honestly, if you want to start a lab. I actually think starting a company is a lot like being a pre-tenure PI. It's a lot like it. That's it. You can find me here. I have a bunch of business cards up front. You can grab one of those too if you want to ping me. So we have 20 minutes to talk about anything you guys want to talk about. Yeah.
So I guess
What's your name? Hold on.
Brian.
Brian.
In the microphone because we're recording.
Okay.
Sina, can you run interference with the microphone? Okay, great.
Okay, so I guess the first question, or what jumps out at me after watching your presentation and what you've done and where you've landed now. So do you have a coding background, or was that something you didn't pick that up at Baylor? How did you land on this project?
Yeah, right. I still can't code. I did a bit of Perl in grad school where I created these awful recursive scripts that would use BLAST as a hacked together genome assembler for one of these weird organisms I was working on. Literally recursive BLAST on Sanger trace reads from a genome project, from an unpublished genome project. The guys at the high-speed computing cluster hated me because I just had these Perl scripts doing this for like days. And it almost worked until I started getting like hippocontigs in my marine invertebrate thing. That wasn't great. And then at the end of grad school, I actually took a Python, or at the end of my postdoc rather, here at Baylor, I took a Python course at Baylor. And for the final project, I actually wrote a useful script. It was about 500 lines of code. When I ran it through a PEP8 tester to see if it met community formatting standards, 480 lines failed. That doesn't mean it didn't work. The code worked. It was just brutally ugly. To do some cloning automation, which kind of harkened back to my first postdoc's focus. Yeah, that's it. I don't know how to code. So everything that I've mentioned to you that sounds like computer talk, I've learned in the last 15 to 18 months. And that's probably most of what I know. You know, I've got sort of a stack of jargon, and I actually do understand what some of it means. But once I'm exhausted, like when I give a talk to serious computer guys or software guys, it doesn't take more than 2 or 3 questions before I'm out of answers. Sort of. Anyway. Yeah.
More questions?
So you were passionate about your coral reefs. So do you still try to like kind of get back to that using your expertise now?
I try to get back to it on the 1 week a year we go on a tropical vacation. My wife dives too, so we actually didn't dive this year on our spring break, but so my kids' school closes for 1 week every March, so we go somewhere tropical. The last few years, though, I've had a week of scuba diving.
Yeah, so it's more like there's like software can help you figure out the signaling and stuff
Yeah,
like that.
it could, and you know, so to answer the question more seriously, I'd love to get to a point where our software is abstracted enough that it would truly work easily for any biological system. So in principle it is. The back end is fairly data agnostic. We joke that we could crunch financial data if we so chose. It's really a general purpose data flow engine. What it would take, of course, is for the user to have an easy way to upload new genomes. Right now we sort of manually push that. So 99% of our collaborators study either human or mouse. Then we have got the dictyostelium folks, and we're bringing E. coli on in the coming weeks. So we're hitting like the major model organisms. It's certainly doable, and probably somewhere like, I'm going to say 6 to 9 months out on our product roadmap to have a way for someone to just upload a different genome. So as long as your organism has a genome build. Alternatively, you could build a pipeline for de novo genome assembly. Again, it's a generic data flow engine. So if there were a good enough bioinformatician working on coral or whatever, they could, on our platform, develop pipelines to do things like genome assembly, or maybe you're going to do a de novo transcript assembly unguided, right, with no genome or something like this. But it's not quite that simple. The software is still sufficiently immature that it takes a lot of manual intervention by our team to kind of make sure the data models look right and get propagated right. So this is stuff that, for example, because I'm not a software guy, completely blindsided me. But it turns out that it's a problem if you want to combine a bunch of data that were even human data, same cells and everything, but some of them were mapped against an ensemble genome build and some mapped against an NCBI genome build. These aren't going to be compatible or easily compatible. So things like data harmonization are a problem even within the same organism. And so to actually build a software that's sufficiently abstract to handle truly any organism, it just takes more time. Now, the next part of that is there a market for it? So on the one hand, I'd love, maybe there is. Maybe this is actually how we build the company, is by getting all the scientists who aren't served by the tools that just do human clinical blah, 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 right? But I can tell you, having worked in those fields, there isn't a lot of money to go around. And academic scientists hate paying for things. No, it's a serious problem. It's one of the biggest challenges of this business. We'd much rather sell to pharma because they are used to buying stuff and they've got deep pockets. In fact, a lot of pharma divisions will buy us and our competitors just to see what works best. Convincing an academic to spend money on software rather than hiring a grad student. Grad students aren't cheap, but the paradigm is I'm going to give this kid who knows a little bit of programming 5 years to figure out how to do something, right? That PIs understand. What, you want me to spend $2,000 a year for a software license? Some of them get it, most don't, or at least they haven't thought about it yet. They hadn't budgeted it into their. So I would love to have some serious like ecological coral reef type stuff going on. There's a huge amount of interest from like the natural products field. Turns out coral reefs make lots of metabolites that could be good at like killing bacteria and other stuff. Maybe we'll get there. We'll see.
Thank you.
So my question is more simply just why biology? Like if you look at things for automating data analysis, you have things like the financial markets, you have actual market research and things like that. Do you see promise in the future with regards to looking at analysis support for biology in this regard?
Yeah. So the bioinformatics market globally is somewhere between $4 and $6 billion this year, and is growing at a 21% compound annual growth rate. So that is a sufficiently large market to justify starting a company and begging venture capitalists for money. It is a really hard market to penetrate for some of the reasons I already mentioned. There isn't necessarily a history of buying stuff. It's a very distributed market. There are a lot of in-house solutions. It's hard to move people off of it. There's a lot of fear over putting data on the cloud, and it's competitive. There are a lot of competitors. So if I were starting a company today and my interest was general purpose, like I'm a good data scientist, where can I go have the biggest impact with data? I'm not sure this is the market I'd enter. But I was recruited halfway into the company's history. They had thought about this. I mean, they had actually given this a lot of consideration because, again, they came from a general purpose data science lab. And a large part of the goal was not just to build a $10 billion company that helps bankers get rich. It was to solve problems in biology. And so for me, this was key. I have no interest in going to work for a fintech company. The business stuff is fun because it's new, but what I like is solving the biological problems. And so one way to think about it, and the way we actually even open our pitch to investors, isn't so much with the market plea, it's with the purpose, the mission. So think about the biggest problems in the world today, right? So climate change is a big one, at least I believe in it. All of the stuff that trickles down from that. So right now we're about to have an unprecedented number of famines in the world. So we desperately need both sustainable commodity production, but we also need more robust crops, crop engineering. Someone over at Methodist had a bacterium that was resistant to all 27 antibiotics they threw at it. This is becoming a serious problem. We need, first of all, to use fewer antibiotics, but we need better ones and new ones. We need much better preventative therapy, right? So or preventative medicine. We've got all of this health expenditure in this country, and our lifespans are pitiful compared to the rest of the developmental world. Yes, we need better medicines, but that's actually not my take. I think we need a better preventative care, and so genomics actually has a real potential to impact preventative care, right? So tailoring lifestyle choices based on susceptibility. And so all of the big problems, in my opinion, that humankind faces besides our own stupidity can be solved with biology. And of course, the problem is that most people working in biology don't have the skills, the know-how, the wherewithal to actually analyze the scale of data needed to solve the problems. And so that's where we're trying to make a difference.
Yeah. A lot of the people in this room whom I know are basic biologists working maybe with the bioinformatics in their lab or some collaborators. So what would be your advice for, even if we are sitting and doing some analysis with the bioinformatics, we can only know some jargons or some kind of p-values, why they chose those, but not necessarily know their pipelines or know the entire flow and their codings and other things. So, but after getting the PhD or master, whatever, if we want to see ourselves working in these kind of jobs, what would be your advice during our 5 years period to develop these skills?
To develop the skills towards being better at bioinformatics? Is that the question?
Yeah, and also know the workflow.
Right. I would become really literate at both Python and R. So how you choose to become literate is up to you. There's a ton of stuff online, Code Academy, Udemy, Coursera. I bet they offer courses here. They definitely offer courses across the street in that direction at Baylor. If you want to really learn it, go across the street to Rice. That's where they teach you how to do coding for real. learn how to program and how to read code. I mean, even if you don't want to be a programmer yourself, you need to learn how to read code a bit. You know, what I think a lot of biologists take for granted is that bioinformatics itself, that's a field of study. It's not just applying some algorithms to some data in some ad hoc way. The number of papers, including my own, that say so-and-so algorithm was run with default parameters is, you know, probably most of them. And so educating yourself about what the tools do and what the flags mean. Biologists are woefully inequipped with statistics. That's another area that, you know, if you understand some statistics, I would still say work with a professional to design your experiments before you analyze them, and then to analyze them. But, you know, these are the kinds of things I wish I knew how to do. This is a do as I say, not as I did sort of lecture. And if you want to become a software developer, you know, again, learn Python, learn R. Python's actually more relevant to software development, but for bioinformatics, they're both really good. And you know, whatever other stack you want to add, and go do it. There's actually now a really, really vibrant tech community trying to grow here in Houston. It's sort of like a seed that's germinating. It's looking for some place to spread its roots. There's a ton of employment opportunity for people who with a bit of coding skill and a lot of passion to learn it. And, you know, frankly, there's a lot. I think there's way more potential in the future for biologists who also know the data side. So, you know, instead of calling yourself a biologist or a bioinformatician, become a computational biologist, whatever that means, where your lab not only generates data, does its own experiments, but is also the key lab in your institute for figuring out how to analyze that data. And then success will come because everyone else will want to work with you. Yeah.
My question is more about the business side. Is there anything that you think you did do or you wish you had done to prepare yourself for being project manager, raising funds, things like that?
This came so far out of the blue that, you know, like this was not something I was planning to do until the day I did it. So I don't know that I could have prepared better. I actually think I did a pretty good job. So I mentioned when I was in the Bay Area, I kind of got this bug for doing something commercial. So the minute that I got to Houston, I already sort of had a foot heading out the door from the lab. I mean, I worked hard in the lab, but I started networking at all the Rice Alliance events, which are venture capital pitch events. I started going to TMCX, which was opening up about the same time. They run a lot of free informational stuff. JLabs does too. So there were these resources that were all kind of converging at the same time, and so I dove into that. This is where I can maybe also make a plug for networking. Seriously, guys, spend some time, use the internet to figure out the difference between a resume and a CV. Build them both, but know the difference. There are lots of cool software products now where all you do is enter the information and they take care of the formatting. So there's no excuse for having something that's ill-formatted, that's ugly, that doesn't have the right stuff. Crowdsource the content. So once you've got a nice draft, literally send it to a dozen friends with a, you know, with a candy bar taped to it and ask them for their help over and over again until you get it right. Not just friends in your industry. Send it to, you know, your college roommate who's now a marketing exec. If your marketing exec friend can read your resume as a biologist, then you've gotten enough of the jargon out of it that it's actually readable by an HR person. Use LinkedIn, but use it well. It's not Facebook, so you're not adding your high school buddies who don't do anything relevant to what you do. You add first people you've actually worked with or interacted with in a professional setting. Second, you add people who offer to connect with you or who you've met in passing who you think will be useful to you. So very Machiavellian, right? Like, you know, for example, a grad student the other day asked to connect with me. I don't know him, but I know that his PI is famous, and I want a reason to have, you know, that connection. So sure, I'll accept your LinkedIn connection. It's really an invaluable tool. And then just network. I mean, it sounds so trite, and a lot of us, like when I go to networking events, after my second or third elevator pitch of explaining who I am, I usually wander off by the food and then go home. But I go and I at least get those 3 introductions, and it's really, really valuable. You'd be amazed how quickly you start, you know, drawing on that network. So those are things I think I did well. Other than that, you know, when I jumped into it, I didn't really know what the startup world was. The kinds of things I thought I had to be concerned about were not the things that we needed to get done first. But there's a great wealth of books and blogs, and so if you think this is something you're going to do, email me, I'll give you a reading list.
More questions? I have two actually. So thank you so much. I really personally enjoyed your presentation. I'm already thinking about having you back in
Sure.
in a few months. I wish there were more students, postdocs using this opportunity. So you mentioned like so many good points. If I want to highlight two of them, when I go to like different programs to present like career planning, career development, and I ask students like, what do you want to be in the next chapter? I was so surprised so many of them wanted to start their own startup. So I really appreciate the last slide you put there from your co-founder, because so many of us tend to get caught into iceberg illusion. So it's not as easy as it sounds. The other point was your last slide and what you do at your current job. One of them was seeking for fund either internally or externally. So many of grad students, postdocs think that if the grantsmanship or grant writing skills is the skill only needed in academia. So again, highlighting what you mentioned, outside of academia, also in so many career paths, you need to improve your grant writing skills, being able to put a good proposal together.
This is actually a key point. So if you were to start a startup, there's a ton of money available from the government. Assuming that really catastrophic things in the funding mechanisms don't happen in the next 4 years or 4 months. There's a ton of money available for science-related startups, not just in biology. And the beauty of this is it's non-dilutive capital. So the problem, if you go to a VC, they might give you $1 million, but they're going to take a quarter of your company. And they're really hard to convince. The success rate with VCs is way lower than the funding rate at the NIH, way lower. Mercury Fund in Houston, which is the most prominent one here, I think they fund 1 to 2 companies out of 3,000 a year that they meet with. So the NIH has a much better funding rate. And it's, like I said, it's non-dilutive capital. They don't take anything from you, and they don't take your IP or your equity. So grantsmanship is a big deal. You in the startup world will be doing a lot of it. You also just write a ton of proposals, and the skills overlap a bit. Even if you're not in the sciences, there are other small business types of things where you're going to have to petition for money. And so, you know, a lot of times grad students, they feel lucky if they're in a well-funded lab where they don't have to engage in this. But, you know, apply for fellowships, apply for grants, ask your PI if you can participate in writing an R01 or something. It'll certainly help you if you stay in academia, because Lord knows you need to do it. But it's important. With that said, I applied. The first thing I did when I joined the company was actually write for an NIH small business grant. And we did really well for our first swing at it, but we didn't get funded. And now we're actually in a weird situation where we can't go back to the NIH because since the corporate acquisition of our subsidiary, we actually are more than 50% owned by Europeans. And so we're not eligible for US federal dollars. But we've raised over $1 million in non-dilutive capital from the European Union through our subsidiary. And we continue to go back to that well.
More questions?
So, let me follow up on the first thing you said about, the startup life being hard. To me, the more and more I think about it, the parallels with non-tenure track faculty are similar. Like all of a sudden you're doing something where your primary concerns are raising money and paying people and promoting your work in some way. So those are all kind of similar. A difference though, in some ways, the startup world is more forgiving. If you're a young PI and you fail to publish or fail to get grants, you're out on your butt, and it's actually going to be hard to get a job at the next institute, especially if you don't keep publishing. In the startup world, yeah, everyone wants to succeed, but it's almost like, you learn a lot from failures, and the number of successful entrepreneurs who have a huge dossier of failed companies under their belt is many. I mean, even famous entrepreneurs, even Steve Jobs failed miserably at the company that he tried to do between Apple and Apple. I mean, it doesn't always work, so. Trying can only help as long as you do have a tolerance for stress and not sleeping and not getting paid much.
Okay, so to my understanding, you're going to stay around?
Yeah, I'm going to stick around for the next hour or so. There was a bunch of food out there that we should probably eat, and I'm happy to talk about anything I've already talked about or other stuff.
So thank you so much for your presentation.